was assistant professor in Colorado University in the US, and he used to work first in Vienna, Austria, and then in the Gleason lab, the US, and as an expert of mosaicism in the brain and in sperm cells. So we're very glad to have you here. For his use.
Perfect. Thank you very much for the introduction. As some people say, unfortunately, I have nothing to disclose. I'm very excited today, and thank you very much for the opportunity to talk about clonal mosaicism in sperm. And so this really is following up on what Anne has just told us about in terms of like thinking about it from a clinical perspective. So I always like to start with this picture here. So I studied neurodevelopment during my PhD, so I'm well aware that this is not an accurate depiction of a sperm cell. However, it is really a nice kind of view of thinking about how the entire blueprint of such a complex organ like the brain is already contained in one sperm cell. And as you all know, one single mutation across the billions of bases is really able to change how the brain develops and cause very impactful human disease. And this is really what motivated us to think about sperm mosaicism. So one thing, this is an educational session, so we hope to have something to offer you, but we also would like to have you tell us something about so that we can learn about your experiences and your thoughts. And so we have this small survey that we put together, and so we would hope that a lot of you are interested in participating, and so I've put this out one here, and we will also have it up at the end of the lecture, so you can either do it during the questions if you're not as interested in what I have to say, or you can move on and later on during the day when you have time, fill in the survey. So for our learning objectives and the outline, really I would like to give you a developmental view of sperm mosaicism, and this is really restating a lot of the principles that Anne already has mentioned before. I will then talk about the patterns of mosaicism across the genome. I will talk a little bit more about the technologies and some considerations when thinking about doing this assessment. I will talk about our work on de novo mutations and going back into the genome of the fathers, and then also about unbiased assessment, both about the results that we obtained, but also some thought experiments of what can we do with this technology. And I will then give a summary that also goes into the practical consequences. So when looking at the basic family unit of a parent and pair and also the children, as you all know, one of the very severe impactful life events for these families is having a child with a congenital disorder. So each situation is very unique and has a lot of unique challenges, but there's one question, as Anne also very nicely pointed out, that is very common for many of these families. And this is, what is the risk of having the exact same disease again in this family? So as we at the European Society of Human Genetics, and we all are geneticists, really we are thinking about it from the genetic perspective and the gene mutation. So during my postdoc, I was dealing a lot with pedigrees like this one here, which is a consanguineous family with 2 heterozygous mutations in one parent each, and if you have the same mutation inherited, you will have a child with the disorder that we were studying. So as we're all geneticists, the recurrence risk here is very easy and very comfortable for us to calculate because it's 25%. It's Mendelian genetics and very straightforward. So when looking at de novo mutation, as we already heard, we now have this event where we think intuitively that there should be no recurrence, but we know that there actually is recurrence from population-based studies. And as mentioned, we normally give a recurrence risk of around 1 to 2%. So one thing that you immediately appreciate, especially after the talk before, is that on the one hand, we have individualized risk that's very precise and very satisfying for us to report. And on the other hand, we have a population risk that's very imprecise and is incredibly frustrating for the families and also the providers. So to think about this in a little bit more detail, we really have to talk about the origin. And so you can see here again, we have a de novo mutation, and very often when talking about these, we assign this very esoteric transmission mutation, where the allele is mutated at the transmission from one generation to the next. But clearly this is not accurate, because conception doesn't happen just magically, but is actually the fusion of an egg and a sperm. So on the one hand, we can have these mutations that really occur at the time of conception, or even later and form these post-sydetic mosaic mutations that Anne mentioned. Or we actually can have them come from the paternal or the maternal germ cells. And as the focus of the session is really on sperm mosaicism, because this is the tissue we can have access to and we can assess, this is what I'm focusing for my talk as well. So now if a mutation is present not only in one sperm cell, but in the progenitor cells, we now have this potential of having recurrence. So, but really to think about this in more detail, we have to take a developmental view of sperm mosaicism and the sperm lineage. And so just to recap, genomic mosaicism, this phenomenon where some but not all cells within a tissue carry a unique mutation, is something that has really emerged throughout the last decades as an important process. So a fascinating insight from the 20th century was that actually every cell in our body has the same genomic complement. So this seems kind of naive for us to not assume so, but back in the day it was really an important insight that every cell, no matter what it looks like, has the same genome. But as we also know, this is not entirely true, because we are all mosaics of a history of developmental and aging mutations. In some cases, these will actually change the phenotype of the cells, and we have this mosaic of phenotypes across our body. So we are very well aware of this in the context of cancer, where a mutation, a somatic mutation, actually causes disease. But so what we are talking today about is what if these mutations happen in the germline and they do not necessarily impact the phenotype of the germ cells themselves, but actually would convey a new phenotype to the next generation. So looking at genomic mosaicism more broadly, we can really distinguish two different types. So one of them is clonal mosaicism, here illustrated by having a 2-cell and 4-cell stage embryo that accumulates mutations that are accumulating in a mother cell, and then they will be transmitted to the daughter cell and to the entire lineage, making all of these cells mosaics of different mutations. So in contrast to this, once we have a postmitotic cell, here illustrated by a beautiful postmitotic neuron that definitely is not proliferating anymore, you can accumulate aging mosaic mutation or aging mutations over time, or private mosaicism. So for our purpose, really, we are interested only in these clonal mosaic events that are also called developmental mosaicism, that are actually happening early on and then get fixed in a larger number of cells. So when thinking about this in the context of sperm, and we're looking at the adult testes with the seminiferous tubules, we now have a population of spermatogonal stem cells here that will undergo spermatogenesis to form sperm in the lumen. But these cells actually come from development, as Anne very nicely pointed out before. And so we can actually spread out the entire lineage in this very simplified model of how development works, where we go from the zygote all the way to the sperm cells, through the primordial germ cells that Anne mentioned before, which are the embryonic cells that will actually form the germ cells both in male and females. So now thinking about the timing of mutations, they can occur very late in the process and will be truly a one-off. They can occur in the spermatogonal stem cells, but because they are only one of many, many spermatogonal stem cells across the testes, again, these are not impactful in terms of recurrence risk. But finally, we have the clonal or developmental mosaic variants that happen either before the split of the primordial germ cell of the soma, from the soma, or after. And now these can occur at high enough rates that they're actually appreciable from bulk sperm analysis, and they also have an important impact on recurrence, and this is the clonal mosaicism that I want to focus on. Just one more picture to kind of put this into more perspective. Starting again here with the zygote, and then having the split from the soma and the primordial germ cells. So if a mutation happens early enough, it will be actually present as this mixed mosaicism that was mentioned before. And if it happens later on, once the primordial germ cells are actually established, you will have a confined mosaicism that will go on and still be present in the somatogonal sperm cells, but not measurable from the soma. So what's quite interesting is that there is a bottleneck event developmentally, where these cells actually origin from maybe 3 to 5 cells, which really can amplify mosaicism that's present specifically in this lineage. And again, these early events are clonal mosaicism that we're dealing with and that can cause recurrence. So we now think about this as like a mutation that arises during an individual's development, and we can make a few predictions based on that. So the first one is just thinking about it in a linear developmental trajectory. Primordial germ cell mosaicism will result to spermatogonal stem cell mosaicism, which will then be appreciated through our analysis of sperm. Because of the processes and because of the way the niche of the spermatogonal stem cells is actually set up, this is actually immutable, so it will not change with age and time. And then finally, this is a prediction that we kind of assume naturally that the abundance, or what I call or will refer to as the allelic fraction, predicts the transmission of the mutations to the next generation. So to think about the patterns, first I want to talk a little bit about the employed technologies. So I do not have to introduce the principle of next generation sequence to anyone here in this audience, but it has a very unique feature that I at least sometimes forgot about before I got into the field of genomic mosaicism. So what sequencing by synthesis of these single clusters of molecules actually allows us to do is to do single molecule analysis. So every read actually is reflective of one molecule. So this means if you have 10% of mutant alleles, you will actually have 10% of these clusters and the reads that represent this. So we can have a very direct analysis of mosaicism just by looking at the individual reads from next generation sequencing. Just thinking about it more from a model perspective, so if we have now a collection of cells that have a reference allele or a mutant allele, we now can think about it doing bulk sequencing analysis, where we are able to count at each position whether or not we have a reference or a mutant allele. And by various ways of mosaicism calling, depending on what you're really looking for, you could then come up with a plot for the allelic fraction, as you can see here. So the allelic fraction here is really the fraction of the mutant allele over the entirety of all alleles that you measure. So a heterozygous variant, for instance, would be at 0.5 allelic fraction, and anything below that would be a mosaic variant. You can do the opposite, where instead of looking at the bulk, you look at single cells. So while this is very powerful for assessing private or aging mosaicism, really it's not as important for what we're talking today about, which is like a more widespread mutation that actually can impact recurrence. And so really what we are dealing with is normally bulk sequencing analysis. So just one last slide on the technologies, and this is not really for us to walk through and really understand all the details, but just to illustrate that what we're doing with mosaicism analysis very often is built on cancer biology and cancer mosaic analysis. However, inherently cancer mutations are different in a few ways. So first of all, we normally do not find mixed mosaicism in cancer, but we have it confined to the tumor. Moreover, very often we're looking at allelic fractions that are much higher than some of the things that we are dealing with in sperm mosaicism. Nevertheless, we do use a lot of these technologies, although they have been adapted over time to be more specific to the questions that we are asking, but they're very prone to analytical errors and also alignment errors or technical errors from the sequencing. So just something to keep in mind when you're thinking about mosaicism analysis, that it can be sometimes a bit more challenging than initially assumed. So just like to summarize this part quickly, so as I said, next generation sequencing really allows the detection of mosaicism natively just by doing the experiment. The sensitivity, as you can probably guess, is related to read depth. So the higher you sequence, the more likely you are to pick up a mosaic variant at a certain allelic fraction, but also you will get more accurate at actually predicting what the real abundance within the sample is. The lower and upper detection limit are determined by errors, technical and analytical, and you read depth. But one thing that we haven't really talked about, but it's also important to consider, is there is this phenomenon of false negative de novo mutations. So this would be a case where actually in the parents, there is a mosaic variant that reaches high enough that it's indistinguishable from a heterozygous variant at the read depth that you're working on. So while the parent might have a milder phenotype or no phenotype because it is mosaic in them, it is actually a germline de novo mutation in the child causing a disease. And finally, an important distinction is whether you are targeting your mosaicism, so whether you know what you're looking for, like for instance for the PrECCare study, where you look at one de novo mutation that you already know was transmitted to the next generation, or whether you want to look at an unbiased view of the entire genome and understand mosaicism at a genomic scale or an exomic scale, or maybe even at a panel sequencing scale. And really, the untargeted detection has some computational challenges that are unique and different from the targeted analysis. So what about looking at de novo mutations at a larger scale? So for this study, we actually obtained samples from 8 fathers. We had matched blood and sperm samples, and we did whole genome sequencing at 200x coverage. So just as a reminder, we typically would look at 30 or maybe 50x for whole genome sequencing. So this is really a 4 to 7 times increase over what we normally do to increase our sensitivity to pick up mosaic variants. So we were very lucky that in our collaboration at UC San Diego, we were able to obtain trio sequencing data, or actually multiple children in some cases. And we looked at all the de novo mutations that were obtained across these 8 individuals, or 8 families. We got the expected around 30 to 96 de novo single nucleotide variants for a total of 912 mutations that we will then assess in the whole genome sequencing data in the blood and the sperm from the paternal side. So in doing this, we found that across these 912 variants, we found 2.5 were mosaic in sperm. And breaking them down further, we found that the majority of them was only found in sperm or was enriched in sperm. And so this goes back to this ascertainment bias because we look at mutations that were actually transmitted to the next generation. So it's likely that we find them at higher levels in the sperm than the blood or exclusively to sperm. We can also do the paternal phasing with long-read sequencing that Anne mentioned before. And when doing this, we were able to phase around 70% of all the de novo mutations, the majority of which was on the paternal haplotype. And you can see across these 481 mutations, 20 or 4% were mosaic. So again, very similar to the numbers that Anne mentioned before in her talk. So when looking at these a little bit more closely at the level of the allelic fraction, again, here on the y-axis, allelic fraction, so what's the abundance of the mutant allele, and we have a rank of all the mosaic variants on the x-axis. And you can see that the allelic fractions in some cases were quite high, so they were close to 0.2 or 20% of all sperm cells, close to 20% of all sperm cells would have this mutation. So you would somehow intuitively think maybe this is really the case only for those that are shared between blood and sperm and are mixed mosaic because they happen earlier. However, we found that in some cases we have either very strong asymmetry or in some cases even exclusivity for very high allelic fraction in sperm that are not present in blood. And I think this is a very important topic that is going throughout both of our talks, that sperm is really the tissue that you have to analyze to get an accurate assessment of this mosaicism. So what about risk stratification? Just using this one data set that I just showed you. So in the red line here in the middle, you have what we would assume would be the risk if we go by population averages, correct? Every single variant has the exact same risk of being transmitted. But if we actually look at it based on the mosaicism level that we observe, we can now see that we can do a risk stratification where some variants have a very high risk of being transmitted, but the vast majority of variants actually has a low risk. So again, really separating into the high-risk mutations and the low-risk mutations from the point of view of having a recurrence or a transmission in the first place. So just to summarize this part, so we found that across neutral mosaicism, so these are just all variants that we found across the genome, 2.5% of de novo single-nucleotide variants or 4% of the paternal were mosaic. We also were able to extend this kind of principle to other classes of variation, in our case structural variants and short tandem repeat, expansions and contractions. As we discussed in the lecture before, pathogenic variants also exhibit mosaicism. So there is this interesting phenomenon that it might potentially be higher than neutral variants. And so this might be because of variability across assessed cohorts, because of intrinsic differences between different classes of disease mutations. But we have some data that also suggests maybe just the exonic regions themselves have a little bit of a higher mutation rate that might also change between different areas of the genome and between different genes. So this is something that's still an active area of research, but something that's quite intriguing just from a basic biological perspective as well. And finally, I really want to stress that we found up to 15% abundance with little to no representation in blood. And so while there is bias because we look at inherited variants, it is very relevant if you look at de novo mutations that are actually found in a child. So what about doing just unbiased assessment altogether? What if we don't have any preconceived notions of which mutations were transmitted or which mutations are present in the next generation? So to really get an understanding of what is like the natural mosaicism that every male runs around with, we assembled this control cohort where we really were interested to think about intersubject variability, intrasubject variability, so sampling multiple sperm samples within one individual and looking at age dependency. To do this, we assembled a cohort of 12 control individuals, which were 18 to 20-year-old undergrad students at the University of California, San Diego campus, who were really excited about the Amazon gift cards that we gave out. And we also had a matched age cohort where we had individuals of 48 years and older. And this was the advanced age cohort AA or the young age cohort YA. So for all of these, we then performed 300x whole genome sequencing, again, even higher than the last experiment that I showed you, because we really wanted to increase our sensitivity for unbiased mosaicism. So we then performed a mosaicism calling analysis, which is shortened here with MSMF, which is really just the initials of the callers that we used as a combination to increase sensitivity and specificity. And this will end up with a mosaic mutation candidate list. So we'll skip over quite a few things that we did with this cohort and quite a few analyses. But I always do like to show one figure that we had in this paper because I think it really summarizes a lot of the things that we found. You can see here in this graph the sperm mosaic variants, so those that are confined to sperm, shared variants, so those that are found in blood and in sperm, and then finally the ones that were specific to blood. So the first thing that most people latch onto, because it's the most obvious effect, is that there is this huge variability in blood mosaic variants. And this is really reflected of a phenomenon called clonal hematopoiesis. And so this is something has been described for aging individuals where individual clones of the blood take over the system. However, the second thing, which was the one that we were most excited about, is really that we have this very stable correlation across age for sperm-specific and shared mosaic variants. So sperm mosaic variants, in contrast to the blood mosaic variants, actually seem to be stable across an individual's life. And we really think that this would suggest that this is a lifelong threat of transmission, and you can measure at any point of a male's life what would be the lifelong transmission risk of mutations. And you can see here another quantification of this where blood is clearly going quite crazy with age, but sperm mosaic variants are stable even across the age groups. So overall, we found that every male has around 30 variants in their sperm that are mosaic and have the potential to be transmitted. We demonstrated that they are stable within 1 year by serial sampling of the same person within 12 months. And we also see that they are stable across age groups. So what, however, is the impact of having these mosaic variants from a population health perspective? So to do this, we did some calculations of how many CL variants, so in CL variants here really define high impact variants that either had a CAT score above 25, which I appreciate is a very coarse measure, but it's like one approach to think about deleteriousness, or a loss of function variant. So this first one shows you that 28 out of 100 males would have one of these high impact variants in any gene. So as you all know, this is not necessarily relevant because not every gene mutation is equal in terms of risk. So if we further intersect these variants with those that are in haploinsufficiency genes, so genes that should have an effect if they are impacted by these mutations, we find it's 1 in 15 males that has one of these predicted potential pathogenic variants in their sperm. And we can further subsect this with different disorders. For example, here we use the Safari gene list for autism spectrum disorders, where 1 in 100 is predicted to have one of these mutations. And we can go back to the allelic fractions that we actually observe, and you can see that for the majority of these males, it would be a allelic fraction of 5% or lower, but we have a good number of individuals that would have very high allelic fractions, and the average comes down to around 5%. So we talked a lot about how measuring sperm is really the key to predict transmission. So one criticism we very often received was, how do you actually know that the mosaicism you measure in sperm is the risk to be in the next generation? To answer this, we actually were very lucky to collaborate with fertility specialists in San Diego. We performed the exact same analysis that I showed you before on 3 additional individuals. And now we also followed up with massive parallel amplicon sequencing, which allows at very high depth to both estimate the exact abundance of the mosaic variants in sperm, but also in parallel, we genotype blastocysts that would have been otherwise discarded and were offered to research to look at the expected and observed transmission. So here we now look at unbiased mosaicism, and we then make a prediction of which variants potentially can be transmitted, and we then test this in the offspring generation. So when doing this across 3 individuals, we were able to detect mosaic variants in each of the 3 fathers, or sperm donors. And then we looked in up to 14 blastocysts, and each of these red squares indicates a transmission event. So we were very excited to see that actually what we predicted somewhat happened, because we do see transmission of mosaic variants that we find in sperm. I want to summarize what we found here on this slide, where we actually think the transmission is indeed predictable. It correlates with abundance, the allelic fraction. So the more mosaicism you have, the more likely you are to transmit a variant. More variants result in more transmissions. And at this point, you might be, wouldn't you have expected this? And yes, we did, but we were very happy that it still happened the way we predicted it. One thing that was kind of surprising is that we found a slight undertransmission compared to our expectation. And so doing some additional modeling, we believe that the most likely explanation is that there's hypermutability of certain lineages. And this is consistent with some more recent work on looking at early developmental mosaicism, where some lineages appear to have higher mutation burdens than others. So one question that I want to pose here, and we will get back to when we're talking about the practical consequences, is this approach desirable? So I'm a basic researcher by training, so I'm very excited about things I can do, but this doesn't really mean that we should do them. And it's something that I think is very important to think about. So before we get to this, however, I want to leave you with a few vignettes and thought experiments to think about in terms of mosaicism. So the first one is, why don't we just do this in blood, correct? Blood is available, blood is easy to access, why don't we just do it in blood? And so here we just did a thought experiment. What if we looked at blood instead of sperm for these 17 individuals? And you can see the false positive rate from blood, so where we would have mosaic mutations that actually do not exist in sperm, and you can see the false negative rate from blood, mosaic mutations that do exist in sperm but were not detected in blood. And you can see the proportion of both is around 0.75, and I think no clinical test that we offer, if we would have a better alternative, would pass the sniff test for being done. So really, and again, I think you're already probably a little bit sick of us saying this, but we should detect mosaic variants from sperm. And this is really, I think, a critical point. And if you don't take away anything else from our session, we hope that's what you will take away today. So what about putting all this information together? And Anna already mentioned this in terms of like putting together these numbers. And so this is a different way to look at it. So on the left here, you have the inherited de novo mutations. So inherited as in they have been present in an egg or a sperm. You have the true de novo mutation that occurred the very first cell division before you have more than one cell. But once we go to the embryo or to a child, we are not able to distinguish those two. What we can do is we can look at the paternal haplotype here in blue and the maternal haplotype here in red. So putting all of these data that Anne mentioned before together, we can think about having 80% de novo mutations being in the paternal haplotype, around 20% being in the maternal haplotype. You can see here these post-conception variants, which will be the same between the two haplotypes. We have oogonial and spermatogonial specific variants, which are unique to both types of inheritance. And then we have the PGC mosaic variants, so in the primordial germ cells. And these are really the clonal mosaic variants. So the first thing you can appreciate is that, indeed, this contribution is the same in the maternal and the paternal side. But because we have more paternal mutations than maternal mutations, once you identify a variant to be on the maternal haplotype, it has a higher recurrence risk, relatively speaking, but not absolute. The other thing that is quite curious is that this block of spermatogonial and also oogonial specific variants actually increases with age. So as a consequence, while the mosaic variants stay the same and are stable across life, you will have this increase of the aging-related mutations. And this conversely also relatively decreases the risk of having a mosaicism causing a de novo mutation with age. So this is somewhat counterintuitive because most people would assume we have more de novo mutation with age. Shouldn't the mosaic variants increase as well? But we have shown that they do not. So they stay stable across your lifetime. So what about mosaicism detection from sperm as a clinical tool there? So we really want to advocate from the PrECCare study and also work that we're doing in my lab and that I've done doing my postdoc, that if we want to think about recurrence risk, to provide accurate familial recurrence risk, I think it's not far-fetched to say that we should really do sperm mosaicism assessment. If you think about mosaicism analysis before conception, it gets a little bit more complicated. So the first part is really something that I think will be addressed very soon. So while there are still technical challenges in detecting mosaic variants in an unbiased manner, I think both the sequencing technologies, the depth that we can reach, and also the computational tools get better and better. So this will not be the bottleneck for any of these analyses. A bigger one, I believe, is the interpretation challenges. So we normally, if we look at the de novo mutation that causes the disease, we use the phenotype of the patient as a very important input into understanding whether or not a mutation is disease-causing. So if we do not have this information, if it's just an isolated mutation in sperm, it will be much harder to assign causative mutation or being a likelihood pathogenic. So while we are having a lot of great efforts, for example, through the MAVES, where we do massive analysis of potential pathogenic variants, this is still something that's much more under development and much more challenging. And then obviously there are the ethical challenges. There is the classical question about what is desirable life, at what point do we have an intervention, and what do we actually want to prevent, or what do we want to encourage. But also the very easy thing to think about is what about accessibility? Who can afford doing these unbiased mosaicism assessments? Will they be paid? And especially coming from the US now, there's a very big issue with the accessibility of healthcare across different populations. And then something we do want to stress is sperm mosaicism is only half of the story. So the maternal contribution really equals the paternal contribution. However, because sperm is accessible, it is a very good first starting point, and because 80% of de novo mutations are on the paternal haplotype, we can provide accurate recurrence risk together with the haplotype phasing that was mentioned before if we do sperm mosaicism analysis. And just as was discussed in the questions, there is the question of accessibility of tissues and whether there's a good proxy tissue for X, but so far we have not really found anything that would correlate more or less. The only thing I would say, especially looking at the aging effects on blood mosaicism, is that probably pure blood cells are one of the more suboptimal choices. Okay, with this I just want to put up the QR code again, and I think it will also be up during the questions. But before we go to this, I also want to have some acknowledgements. I want to thank my postdoctoral lab, where a lot of these projects were started. My fantastic colleague, Shashu Yang, who was an integral part to a lot of the work that we did, our collaborators and the participating individuals and families, as well as our funding. And I'll put up the QR code for you again, and I'm very happy to have any discussions, thoughts that you have, or questions.
Thank you very much for this wonderful talk. Are there any questions in the audience first, maybe? So we'll start with the online questions. So we have Lauren asking asking, can you speculate whether clonal mosaicism in sperm has changed since the last century and the preceding centuries?
Yeah, I think that's a very fascinating question. So thinking about it in terms of what can impact early mosaicism in development. So I think there have been a lot of thoughts that maybe environmental factors could really play a large influence, but as Anne actually pointed out, we do not really find that there's a huge variability in the number of de novo mutations if you think about it in terms of orders of magnitude. So while it is possible that things like environmental toxins do increase those mosaic variants, it seems to be not at least a very clear-cut effect. So I think the biggest impact so far that has been described are these very specific mutations that are, in terms of genome maintenance and repair. And I think the one case that is not genetic was due to chemotherapy. So that's like the one big one that we can identify, that if you have chemotherapy, you will get more mutations, but that's not necessarily the developmental, but like the later ones. So it would have to be chemotherapy during pregnancy, which, yeah.
Question in front?
Well, it follows this one. So the way we are talking to our patients about the age of the father to some of these diseases and the mutations in the sperm, so it's wrong. So it does not really increase.
So the number of de novo mutation does increase with age. So you will have a higher risk if you are aged as a father to transmit a de novo mutation, and because de novo mutations have always this potential of hitting a gene and causing a mutation, this is definitely true. So the older a father, the more de novo mutation you will have. What, however, somewhat ironically does not increase is the recurrence risk, because you have this very large basket of de novo mutations that definitely have no recurrence risk that accumulate with age. And they basically dilute out the mosaic variants that are present already from birth, if that makes sense. So there's a higher risk for aged fathers,
Not for the recurrence.
but not the recurrence risk necessarily. Yeah. But again, I would not advocate for not testing just because a dad is aged. I think if you're able to do sperm mosaicism testing, I think it's adequate across the age span.
Thank you. We'll move to the back and then go to the front. Please introduce yourself and ask your questions.
Hi, my name is Ava Oberleck from Technical University of Munich. Thank you for your talk. At our institute, at least, we haven't implemented sperm testing yet for mosaicism. And I recently asked the director of a large lab in Munich, and she also said that they weren't doing it yet. Maybe you can give us an idea about how like the actual clinical appliances of this, maybe in the US or in other countries that you know of?
Yeah, so I think this is a great question, and to my frustration, because I have to write in every IRB application that I will try to go through a clinical lab for cases that we identify, we have not identified a lab that is yet offering it in a CLIA certified manner, which would be the genetic testing for the US. I think just technically speaking from the lab kind of side, the extraction of sperm has some challenges, but it's not too difficult, quite honestly. So it is easily accessible. And then the biggest question is like, how do you implement targeted amplicon sequencing? And I think one solution that people have proposed is just going for deep exome or deep panel sequencing as an alternative because it's more easily validated and verified. But yeah, I think it's just a matter of willingness of labs to take on this challenge of extracting the sample and doing the customized test or doing a panel test moving forward. But yeah, unfortunately, we are not aware of any labs so far that do offer it routinely, but I think it's probably only a matter of time. And I think studies like ANS really push this forward in terms of like, this should be something that is available to patients that have a de novo mutation.
Thank you.
Thank you. The front.
Hi, Edith Konen, Maastricht, the Netherlands. Thank you very much for your talk. From a reproductive perspective, you said that, and which is obvious, sounds obvious, that you see an effect of chemotherapy. Now, in general, we tend to wait 2 years before we start reusing fresh semen in IVF. Do you think that's an appropriate period? Have you looked at that? How long does it last, the effect?
So I think for the chemotherapy, the impact is really more on the individual sperm cell, which you don't necessarily have the ability to test. It's kind of like Schrödinger's cat, that if you test the sperm cell correct, you can't use it for IVF anymore. For the mosaic variants, I think the chemotherapy for an adult male would not have an impact directly on sperm, but it would have an impact on the overall mutational landscape. So I think for that, it's definitely, I'm actually not sure, and Anne might know this, whether anyone has looked at the long-term effects of chemotherapy on the spermatogonal stem cells. And that's an interesting question, but I'm not aware that anyone has looked at it directly.
We have a question also online that says, could fibroblast tissue from the umbilical region be a better source of detective of mosaicism in view of migration of gonadal cells through visceral yolk sac during very early embryonic development?
So the question is about embryonic cells from a father, so essentially banking those cells at birth and then doing the mosaicism analysis. I think just lineage-wise, you would not expect that those cells are better, but it is possible. I don't think so, though. I think because of the very early separation, while blood has its issues, I think these very early mosaic variants are normally pretty well represented until you have a massive clonal collapse, which you normally only would expect at 60 years and older.
You would find it in other tissues from the...
Yeah, I don't think you would have like better yield from the umbilical tissues, if I understood the question correct. But it is possible. So it's also something that I think we still want to test more systematically, because I believe there might be tissues that have maybe better representation because they get fixed early mosaicism. But so far we haven't identified anything that would be able to distinguish those things better than like any other somatic tissue.
Another question in the front.
Maris Laan from University of Tartu, Estonia. You spoke about the gonadal somatic mosaicism, which is linked to development disorders.
Hmm?
My question is, has anybody analyzed, is there any group of somatic gonadal mutations that will be linked to low sperm counts, mainly fertility?
I do not think that anyone has looked at this specific. I think your point is well taken, correct? If you would have a mosaic mutation that is abundant enough and actually causes like sperm defects, it is very possible it would reduce the sperm count. The question is whether such a mutation could be impactful enough, because if you're thinking about it, the highest you would have that is non-germline is that half of the cells carry a mutation, so a 25% allelic fraction. So you might have a reduction up to half of the sperm count. Whether or not that would be really relevant for most individuals, I'm not sure. So I think if you would already have a borderline sperm count, and on top of this you would have a mosaic mutation that causes such a defect, conceivably that could be an important contributor to infertility. But so far we haven't found any mutations like that. But as for most of genetics, probably they're out there and we just haven't looked hard enough.
Just because it may be related to that, we have one more online question from Yuval. And he asks whether, I think he means cases where you have surprisingly high mosaicism in sperm, that you had a chance to look at morphology or functions, or potentially even locomotion in carrier in individuals that have very, very high levels of mosaicism?
Yeah, so the only test that we are doing normally is like very coarse morphological assessment, so we know whether it looks like a sperm cell and has a tail. That's pretty much where our IVF knowledge ends. Yeah, so we haven't looked at any functional features. I think though, for like the surprisingly high mosaicism that I mentioned, if that's what the question refers to, so we think this is really a function of normal development, because you have this bottleneck of variants, and so this really amplifies mosaicism in the germ cell lineage versus it stays kind of relatively low in the blood lineage. So we don't think this is necessarily an impact on the sperm, but it's just like a function of the developmental lineages and bottlenecks.
Thank you. I see 2 more questions at the back.
Hello, my name is Nikolai Kohlschmidt from Bonn in Germany, and thank you very much for the presentation. I'm still a little confused. Maybe I've misunderstood it. From your data, I got that sperm of young students has the same mutation rate or frequency as the sperm of elderly male faculty. On the other hand, you confirmed that there is an age risk for mutations, so paternal age risk. But how do we bring these two things together? Or did I misunderstand?
No, I think this is a point of confusion that we hear a lot of times. So I think the point that we really want to make correct is that across the de novo mutations, so if you would look at offspring and you look at, for example, 100 de novo mutations, we would assume that 4% of these are actually already mosaic in the parents. And so those 4% do not change with age. So the rest of the de novo mutations, however, of these 100, they actually are the ones that are variable with age. So a young father might have 40 of those, and an older father might have 80 of those, but the mosaic ones stay the same across the age.
But I mean, if you find a mutation in sperm, there is a mutation, and if an elderly father reproduces, it may be transferred to a child. So we should actually correlate. I mean, what you find in an ejaculation, a sperm sample should actually be transmitted.
Yeah, so I think the point here that I have to make, and that I might have not been very clear about, is that we are talking about detectable mosaic variants, correct? So we are limited by our read depth, what we can detect, and that's also like what limits kind of like the relevance, because if something is not even detectable with our very sensitive methods, it does not have recurrence risk. So the thing is that those mutations that increase with age are not detectable with these methods. So they are not detectable with mosaicism analysis because they did not happen early enough in development. And so they are actually the bulk of the de novo mutations that you will see in the next generation, if that makes sense. But I'm very happy to discuss more. I'm happy to talk about sperm mosaicism all day if you want to find me after.
We'll move to the last question, please.
Hi, mine should be pretty quick. My name is Lauren. I'm a clinical geneticist in Canada. I was just wondering if there is any sort of established list or database for those conditions like SCN1A where there does tend to be that higher rate of either somatic or germline mosaicism. Just from we kind of end up looking it up on a case-by-case basis a lot of the time, but I was wondering if there's any kind of curated list or anything somewhere that can kind of help point clinicians in the right direction as to which genes to worry about that may have that higher risk.
Yeah, I was just looking at Anne to make sure, and she was shaking her head as well. So I'm not aware of any. I think it's a good idea. The problem at the moment is that I think most of these are, they are big cohorts in terms of like doing the actual research and getting the samples, but they are small cohorts in terms of getting really high accuracy of what the recurrence is. So we have thought about ideas where we could approach it more from a basic perspective. So what if we actually look for this systematically across the genome for mutations that may cause changes in selection in vitro systems that could help guide these kind of things. But we haven't done so yet. It might be a good idea for review, and I'm looking at Anne again, to get these lists of genes and quantify this better. Thank you.
Use your phone and scan. I think Anne and Martin will take the feedback very serious, and if we all encourage them, then there may be a database. So scan.
Yeah, I'm afraid we'll have to close the session. Thank you very much to both speakers.